Thank you so much for the invitation. I will try to provide a crash course on the development of probiotics from the lab to the real world, the things that we've been doing. And this is like covering everything since we started the concept and how we selected these framework and the first tests and the field work very quickly. And of course, most of it's published. So I will present some new things that are not, but anything else, people can just reach out to me and I can explain the details. So first of all, which is something we all know, healthy microbiomes, it's equals healthy organisms, healthy ecosystems, and healthy humans. But of course, what's happening in our world is that degraded ecosystems, they tend to move the beneficial microbes and the assemblages towards a more pathogenic assemblage. So that happens, for example, for corals, for humans, especially talking about corals. When corals are exposed to stress, the beneficial microbes that are so important for their health, they are replaced by pathogens. So now corals have a dual problem. They don't have the beneficial microbes that they rely on, and with them the nutrients and the control of pathogens, the degradation of toxic compounds, and on top of that they need to deal with the pathogens that overgrow because they are more adapted to these stressful conditions. So first thing, considering the vote, is that the beneficial microbiome needs to be preserved and needs to be stored, because in the ocean we've been seeing a replacement where these beneficial microbes are being overgrown by pathogens. And this is one of the consequences, coral bleaching, which is the first step before corals are completely dead. We had massive heat waves hitting the entire planet in the last 2 years. Some coral reefs completely dead in some locations. And basically what happens during this process that is especially driven by increased temperature is that the symbiotic relationship between algae and corals, the photosynthetic algae living within corals, is disrupted because they start to compete. They start to starve and start to compete for resources. Then eventually the corals expel the algae that goes away with most of the pigments. This is why they look white, because the tissue is transparent mostly, and then you can see the skeleton when you don't have the algae anymore. But together with the algae, the most of the energy, about 80% of the energy that corals need, is lost and part of the microbiome. So these corals are starving when they are bleached, and they are about to die if the temperature doesn't go back to normal and the algae returns when the corals are still alive. And so what we thought was that we could use probiotics to keep these corals alive while they are bleached, because of course these other microorganisms, in addition to the algae, can also provide lots of nutrients. They can control pathogens during this process. They can keep the algae as well and provide nutrients for the algae, and therefore they could be buffers to keep these corals alive, like medicine for corals while the corals are bleached. And so in 4 or 8 weeks, when the temperature is back to normal, the algae will find a living host, and they will return to the symbiotic mutualistic relationship. So we came up with this idea. We made a parallel with plants and humans, thinking about the coral physiology, the things that the microbes could contribute with, considering what happens when corals are stressed. So when corals are stressed, there is an increased production of reactive oxygen species, DMSP, a decrease in the availability of nutrients. So we came up with a framework that could indicate the microbes within the coral microbiome that live there but are lost, that are also contributing to these beneficial, at the time, potentially beneficial mechanisms. So we could give nutrients, we could degrade toxic compounds, we could control pathogens if we could restore this microbiome. So basically we go to the field, we take the corals, we isolate the good bacteria, we keep them in the lab. When the corals are stressed, we go back, we apply this bacteria back in higher months, and with that we keep these corals alive. When the temperature goes back to normal, we don't need to do anything because these corals will survive. So we are basically stocking good bacteria in the lab and restoring them when corals need them. We tested that across. We proved the concept that could actually provide protection for corals in tanks and mitigate bleaching. Then we proved the concept that in tanks, we could prevent mortality, reduce mortality, and that was correlating with coral health proxies. In this case, the photosynthetic capacity of the associated algae, which is a proxy for coral health, and it's called FEFM. And we can see that when corals are maintained at normal temperature, these levels are very high. When corals are exposed to stress, these levels decreased, and they were exposed to stress but treated with probiotics. Even if these healthy proxies and the coral decreases and corals are bleached, they recover and they go back to where more or less what they were before, and no mortality is observed in all of our experiments. And at the same time, by running all of these experiments several times using several different hosts, coral species, several different microbial assemblages, we also managed to describe some of the underlying mechanisms protecting these corals, because we could see, comparing corals that were exposed to stress and protected with probiotics versus the ones that were treated with placebo, we could describe some of the post-heat stress disorders that happen, that are triggered by stress, that are not mitigated when the placebo is applied. We could help corals evade mortality, and we could also describe and validate some of the mechanisms that we proposed as beneficial. For example, in this case, we selected one bacteria that was capable of degrading the MSP, which is a compound that is normally found in the coral holobiont, but it's increased in abundance when corals are stressed, and that can attract pathogens and can cause toxic reactions. So we thought that degrading it could provide nutrients and at the same time mitigate the problem. We selected this bacteria. We saw this bacteria was being enriched when this probiotic was applied, and we saw in the metabolomics that we had indeed a decrease in the MSP concentrations when we had the probiotics. So with that, we could validate and correlate these with coral health. And across, we run several different experiments to test the application of probiotics to promote coral growth, and also other groups ran some experiments as well. And we've seen that it's always beneficial to mitigate toxic effects of other pollutants, to control pathogens, to mitigate heat stress, and to promote coral growth across different species, combinations of microbes, and laboratories. And with that, we thought, well, now it's time to go to the reef and try it in the real world. After so many experiments, so many years, we established this underwater city here in Kaust, where we have a very well-controlled area where we started to run not only the experiments, but the risk assessments of using probiotics in the reef, trying to see what was the response of the entire ecosystem. So other organisms, water, sediment. And we started doing this like that, applying in syringes, which is not scalable, but it's a way to start experiments in a very well-controlled way. And we've been expanding, developing devices that can be used underwater. So we have containers, we can put the probiotics, and automatically the hoses can go all over the reef. This is a prototype, and inoculate this for us. We can also use beads that are very common in agriculture, alginate beads of slow release. We can put the probiotic, some devices with these beads, or spread the beads, and they will be slowly releasing probiotics in the reef. And with that, we have a way to scale these experiments. And we've been running field experiments for the last 4 years and a half, different experiments, different coral species, different delivery methods, and different approaches. Some of the results are already out. Some of them are published with showing that there is no harm for the other organisms, the environment and other organisms, and also that these probiotics can be incorporated, and some are coming out now. One of them is being submitted this week, showing that we had improvements in coral health when we have probiotics applied in the field, not only during a mild heat wave, but also during a massive heat wave. And all of these experiments are coming out to show that coral probiotics are not causing any harm to other organisms, actually even improving, restructuring the microbiome in a potentially beneficial way, because we see reduction of pathogens in pathogen abundance in fish. We see improvements in sponges and restructuring some potential beneficial microbes. And we've been seeing also improvements in the overall biodiversity of the reef, where we saw after the heat wave of 2023, we had these structures where we have a mini cosms of microcosms of the diversity of the reef. And we saw that after the heat wave. We used this for risk assessment, but the results were even better because we saw that after the heat wave, we had decrease of 80%. We had mortality of 80% of the overall diversity in patches around placebo corals, and only 40% reduction. So we still had 60% versus 20% of the overall diversity when probiotics were applied. And also we've been using this for restoration and increasing significantly the survivorship of outplanted corals in restoration efforts, so that we are expanding and trying these microbial therapies for other hosts and applications. And of course, as Maria de Gloria said, we've been therefore advocating for the inclusion of microbes as a group that needs to be protected and harnessed to protect other organisms, because we need to preserve these beneficial microbes. As I showed you, they are also sensitive. They can also be threatened. And of course, we can use them to protect other organisms, for example, corals. But there's no magic pill, right? It's one of the things we have to do. We have to mitigate the causes. We have to integrate it with other tools and use a multi-trophic and multidisciplinary approach. With that, I thank you. I thank my team, the heroes behind the generation of all of this data, and I thank you all for your attention.